Now, the genome, different genome of different organisms, they contain a certain motif. What are the motifs? Motifs are certain organized pattern of the sequences or all that. Now, the finding regulatory motif in the mammalian genes is another important subject which we will study in this module and find out that why it is so important to find out the regulatory motifs in that genome. Now, what happened? In 2005, Eric Lander and Manolis Callas and their team, they reported finding regulatory motives. And how they found the regulatory motives? By bioinformatic approach. So you see, the bioinformatic approach now is being used to determine the regulatory motifs which are present in the sequences of the different DNAs. Now these guys, they reason that regulatory motifs and the regulatory motif are 6 to 10 base pair long. They are only small. They are just like small sentences or small, very small sentence which control the whole text. And they are most likely to be found in the upstream regulatory region of gene where transcription factor are likely to bind. So these motifs, because they are regulatory motifs, where they are to be found? They are to be found in the upstream region of the gene. And in the presence of that, in their neighborhood, the transcription factor, they are also present. So this is how, with the transcription factor, they join and control or regulate. Now, in the 3' untranslated region, we know there are certain untranslated regions present in the gene, and that is toward the 3' side of messenger RNA and other regulatory molecules that bind and regulate the messenger RNA stability and translatability. So in the 3' untranslated regions of the gene, where messenger RNA and other regulatory molecules, they are bound, they are attached and regulate the messenger RNA stability. Now these scientists, they further reasoned that regulatory motifs are likely to be conserved among related organisms. What does it mean? That these regulatory motifs, which are very small, they are likely to be conserved. They are not changing from one generation to another or all that, and they will be conserved among the related organism in the species. So they compared human, mouse, rat, and dog. So they took human, mouse, rat, and dog genome to find out the conserved sequences in the 5' flanking region and in the 3' untranslated region of the gene. So these are the two ends which are important, where these genes or where these conserved sequences are likely to be present. So they took human, mouse, rat, and dog, 4 different species. Now these researches, they focused about 17,000 genes. 17,000 genes in the 4 species that I have indicated earlier, and they were well annotated. They were well annotated means that all these 4 species, they were well studied. All those genes were well discussed, known in detail, labeled, and studied. Now, the author defined conservation as follows. How we define that this part of that is conserved? A conserved occurrence is a motif that is absolutely conserved in all 4 species. That is, there should be a pattern of sequence, small sequence, that should occur in all the 4 species, which are so different, what to say. That is, a man, rat, dog, and all that. And we should find that there should be present, absolutely conserved in all these species when we study it. Now, another, this discuss that the conservation rate is the ratio of conserved occurrence of a motif to the total occurrence of that motif in the part of human genome under studies, that is the promoter region, for example, because they will be near the promoter region. So that will be the conservation rate. And the conservation rate is ratio of conserved occurrence of a motif to the total occurrence of that motif in the part of human genome. That is, they took the human genome, find out that motif, its occurrence, and the ratio was that. Finally, the motif conservation score, or MCS, is the number of standard deviation, which is a statistically derived fact. Now, the summary of this is that by using computational biology techniques, Lander and Callas have discovered highly conserved sequence motif in the promoter region and 3' untranslated region of the 4 mammalian species, including human. So they found in human as well. The motif in the promoter region probably represent binding site of transcription factor. That is the transcription factor, that's the binding site, and most of the motifs in the 3' UTRs probably represent binding site for the messenger RNA, which is involved in the discussion of the protein. So we saw that how the bioinformatics has helped us to understand the conservation of different motifs in the cells.